Welcome to Unchatted Waters. In this episode, we'll serve up a twist on the British classic, exploring how fish and the electronic microchips found in our tags can tell us where fish go and give an insight into population movements and migration. So grab your salt and vinegar and let's take a bite into this fascinating topic about fish and chips. Welcome to Unchatted Waters, the podcast that talks to the people behind the science. I'm Kirsty, your host, and I'm so excited to introduce my two guests. Serena Wright is a fisheries scientist specialising in tropical tunas and working in overseas territories. She explores what fish do and where they go using electronic tag technology. Jim Ellis is a fisheries ecologist specialising in sharks, skates and rays. He's been working on fish biology and how fish use and interact with their environment, and he's been involved in scientific surveys for nearly 30 years. Welcome to the podcast, both.
Hello.
So nice to have you.
Lovely to be here, Kirsty.
Great to be here.
So I'm going to start with you, Jim. I want to ask, why do we need to know about fish populations?
Well, fish are very important parts of the marine ecosystem. A lot of species are exploited for food or fish meal, and they're as one of the most diverse groups of vertebrate around the British Isles. They're also incredibly important parts of our biodiversity and in the structure and function of all our marine ecosystems.
So as well as fish populations, what else do we need to know about fish?
There's a range of things we'd like to know or need to know about fish. Where they live, how they move, how they reproduce, what they feed on and their role in the food web, and also when we want to assess populations, how we might identify what are discrete populations to examine.
What's a discrete population? The jar alarm's gone off there.
Oh, okay. So basically, within the global population of a species, they might form discrete stocks or subpopulations that are discrete and more or less breed and reproduce independently of other stocks. So they're pretty much got a self-contained population. What we would term as immigration or emigration is minimal, and how they reproduce and die affects the population level.
So how do these populations change over time? What affects how they change?
Well, probably two of the most important factors are predation, or natural mortality, and also fishing mortality. So how many of the fish die. Then conversely, how many coming into the population, so how they reproduce. So for example, a species such as cod can shed millions of eggs, and in certain years there'll be a really high recruitment because the environmental conditions have been just right, and a lot of the eggs and larvae will become juvenile cod. Other species might have only produce a few young, and so the numbers of recruits that come into the population is much more dependent on the population size of the breeding females. The predator-prey interactions and reproductive output are key factors determining the numbers of fish in a population, but so are environmental conditions. So obviously many fish, they prefer waters of a certain temperature, salinity. Some prefer more oceanic waters, some more estuarine waters.
Serena, I was just going to ask you, in terms of understanding some of those parameters, how do we measure things like population structures that Jim's talked about there, such as age and growth and reproduction on a practical level? How do we do that?
It's a good question. So to get a good understanding of the reproductive output of your fish populations, you need to understand how reproduction changes with size. It might be that your older fish have better quality eggs, which are more likely to survive. It's also important to understand what size your fish become mature and start producing viable eggs. But it's also important to understand where they're going to spawn. If that changes throughout the years, is it linked to the environment, as Jim was talking about? So there's lots of different ways of exploring how fish reproduce. Just basic market sampling of lengths, taking ear bones, so the otoliths, which show you how old your fish is, will help to better understand some of these basic parameters.
So in terms of what you've talked about there, in terms of sampling dead fish, how can we understand where live fish go and what they do?
An important tool that we often use is tagging. So we can use tags, so little plastic identity tags with a unique number on to show where fish are when they're released and where they're recovered, so movement from point A to point B, but also how fast they've grown within that time, what the condition is of the fish when they're released and recovered, and maybe that's affected by the environment. So you can look at, the temperature experience, if you've got satellite derived data and things like that.
Right, it's like putting a name tag on a fish, basically giving it a name and knowing who it is. So how long have we been doing this mark? We give them at release and then recapture, so mark, recapture tagging, Jim.
Well, some of the earliest tagging of fish was actually undertaken in Scotland in the late 1800s. To start off with, because salmon have what's called an adipose fin, and that means its second dorsal fin is very, very small and fatty. And what some early scientists and naturalists would do would cut off the adipose fin, and then they would know how many fish, if you cut off the fin of a small salmonid, does it come back to your river? Now, obviously, that approach has ethical considerations, but also it doesn't tell you the exact nature of when that fish was marked. Since the late 1800s and early 1900s, various other methods were developed, primarily involving attaching a numbered disc to an individual fish. There are various ways you can mark a fish using a tag. So this would be a small disc. In the early days, they would have maybe made of vulcanized rubber, things like this. Nowadays, it would be plastic. But with each tag having an individual number to relate to the fish being released. Now, these tags can be attached to the fish. Early studies would sometimes use needle and thread. Other times it would be a metal pin just to secure it to the fish. So much like having your ears pierced.
I'm going to think of my pierced ears in the same way again.
So the beauty of this approach is it enabled early scientists to tag and release large numbers of fish, primarily things like scapes and rays, certain flatfish. And by releasing large numbers of fish with these individual tags, they could work out, first of all, where fish moved. A lot of the early investigations were undertaken in the North Sea, so they could follow the nursery grounds through to offshore grounds where they would feed and breed. It would help understand the longevity of fish. So the longest time you have a fish at liberty is an indication of how long they can live.
Brilliant. So we've talked a little bit about the history of tags there. How is that different to how we tag now?
There have been some big changes in how we tag fish in the wild in lab settings. A lot of the developments have been on the materials, as Jim mentioned, and moved to more plastic identifiers and actually changing the physical size and structure of the tag. So maybe discs, more for ray species or sharks, and more kind of like a spaghetti style tag for your more round fish like your salmon and your tunas. So I think big changes have happened really for the conventional tags, but also we've started to use more complicated tags like electronic tags, which allow us to get a lot more information for the fish from the point of release to the point of recovery. So, I mean, some of the main developments that have happened have been on what data is recorded. So initially, maybe there'd be a few thousand points of data that you could record a few decades ago, maybe 30 years ago. And now we can record millions of data points from the point of release to recovery. We've changed from maybe just recording pressure to understand the depth use, to including things like temperature, light levels, activity in the form of acceleration, but lots of other metrics that may be useful to understand where fish are, what they're doing, and why.
Thank you so much. So we've had a little bit of information about the types of tags that we have in terms of technology and the physicality of those tags. So I want to get to the basics a little bit more and say, what was it like when you first tagged a fish? Can you describe it for me?
Yeah, well, the first fish I tagged would have been a greater spotted dogfish or a bullhuss. And we started tagging these during our groundfish surveys and beach trawl surveys in the early 2000s. We often would release these fish after them being measured, and we thought this is a good opportunity to better understand the ecology of some of these fish.
Sorry, the German alarm went off there. Could you explain what ecology means, please?
The ecology of fish is basically what they're doing, how they interact with their environment, where they go. So it encompasses just their biology and what they do in relation to the environment and other species.
So continue talking about your greater spotted dogfish.
Okay. Greater spotted dogfish are a wonderful species. They can attain a length of about 120 centimetres. They're a typical kind of small shark that lives close to the seafloor. Not your typical shark that you'll see on television, but a very, very endearing type of fish. Very, very thick skin. So actually using a Peterson disc putting the Peterson disc wire through the dorsal fin, a simple technique to effectively secure a tag to the fish. But obviously it takes a little bit of effort given the thickness of the skin.
Thank you. And I'm going to reflect that question onto you, Serena, as well. Tell me about the first fish that you ever tagged.
The first fish I tagged would have been part of my PhD, and it would have been a cod, and that would have been the Raceways, which was a facility allowed us to do experiments on fish before they went to the wild, or to understand things like their physiology and behaviour in captivity. It was quite a large cod from what I remember, and I remember it was quite tricky to anaesthetise and make sure the fish was in a good state when we released it, but I had some people with a lot of experience teaching me, so it was quite a nice experience to have that as my first fish, because I could actually watch it as it recovered and as it got used to having the tag attached. So yeah, that was my first experience.
So that was both really nice personal stories about some fish that you've tagged. So can anybody tag a fish?
Well, no. So, I mean, it depends on the tags that you're using, but for electronic tags, there's a lot of training, not only for the individual that has to deploy the tag, but also to understand what the most appropriate tag technique and location should be for that individual. So there's a lot of work that has to happen before you go out and do the electronic tagging, before you can go out in the field, for example, and put a tag inside or on the outside of a fish.
So in terms of location, are you talking about location on the fish or location in the environment, or both?
I'm talking about location on the fish. So it's very important to understand how long that tag's got to be on for. You make decisions on, if it's a long deployment, it may be better to go internal because you have less issues with growth and drag. If it's a relatively short deployment, you could go with an attachment technique that would allow it to pop off or detach in a different way. So the physical location and tag type is quite important to consider before you start any tagging experiments in the field.
In terms of non-electronic tags, the typical mark recapture tags, it's very important to recognise that there is a high degree of competence that's needed to, first of all, capture and handle the fish appropriately, and to apply the tag as effectively as possible to minimise stress on the fish, potential for any infection, and to ensure that that fish can be returned back to the sea in as good a condition as you caught it. And there's a lot of fundamental training that's required to allow people to be able to do that kind of work. I think it's also important to note that tagging fish also requires the data. So actually making sure that people are fully trained on how to record the length, sex if it's appropriate for that fish, its vitality, and where it's being released.
Sorry, the jargon alarm again. Could you explain vitality for me?
Oh, just how lively it is, essentially. Is it a very lively individual or is it sluggish? How is it moving? And how you think it's... Yeah. So actually ensuring that the data are available and stored correctly to where the fish has been released is fundamental. It's also very important to use the right type of tag for the species and to also make sure there's recapture information.
Brilliant. So you talked a little bit about competence there. So do you need a special license to tag? Do you need to be signed off by anybody? How do you judge that competence?
To allow you to undertake tagging work in UK waters, there are lots of rules and requirements that are dependent on the tag type. For example, electronic tagging requires both a personal license and a project license, but all of those you have to get through the appropriate authorities.
So we've talked a little bit about data. Jim, you mentioned a few aspects to the data during deployment. So I wanted to ask you, how do we get data back from tags?
This depends on the type of tag, but in general terms, a lot of the mark recapture tags and some of the electronic tags come back from fishermen, mostly commercial fishermen, but occasionally recreational anglers as well, landing a fish which has a tag. They can get a reward, and most of those will come through various port offices. The details through the authorities at the ports will come to relevant fisheries laboratory. Good practice when you're tagging the fish is to have some form of communication, some posters, flyers, letting the local fishing community know that a particular species is being tagged in that area, so they look out for those tagged fish. There's also other different types of tag which have then different types of efforts to recover the tag and the data. So for example, when in the past when people have done a lot of tagging mackerel, that would usually be a coded tag gently inserted under the skin, and so the factories processing mackerel would have a scanner to look for these tags. Some of the electronic tags which can pop off, and they have a buoyant jacket, so then they'll be washed ashore, and then any beachcomber can find these tags, send them back to the laboratory and get a reward.
So they just need to look at the outside of the tag for all of those details.
Yeah.
Where to return it and what to do
Yes.
if they find one.
And then in terms of returning the tag, there's usually a contact telephone number or a website. So for example, here at CFAS, we have a whole web page dedicated to how to report a tagged fish.
So Serena, do we always need to get the tag back to download the data?
Some of the most expensive tags that are available, satellite tags. When the tags detach from the fish, they start to send data via the satellites that are overhead so that you physically get it back at your computer without ever needing to see the tag from beach recoveries or the fisheries. Depending on the size of fish you're working with, there are different options. So for the tunas, for example, which can migrate thousands of kilometres, then you need to be able to get those back without requiring it to be collected from a beachcomber or a fishery.
But if you do find a satellite tag, still return it.
Yes.
Because they're very expensive, aren't they?
And the
good rewards.
Yeah, and if you do recover a satellite tag, you get way better quality information than if you're reliant on the satellites themselves. So
just an option in those oceans that you can't necessarily get to.
Exactly.
Okay, so we've talked about the types of tags, we've talked about deployment, we've talked about returns. So let's talk about data. What can it actually tell us? Once we've got this data back to our computer, what can it tell us about fish movements, Jim?
Well, it tells us a whole range of things. The earliest mark recapture tagging experiments tended to be focused on where do fish move. So you release a fish at point A and it's recaptured at point B. So that means we know that fish has moved that minimum distance over a particular window of time. Through that, then we can understand the dynamics of the stock. If we've tagged a lot of fish, then it's possible to get a much better understanding of how they grow. It will tell us a little bit about the longevity. When we analyse some of our data for skates and rays, some of them had been at liberty for 16 years. It also allows us to better understand the mixing between potential stocks of fish. So, for example, if we have a species in the North Sea and in the Eastern English Channel, and we want to know if they're linked, by tagging fish in one or both areas and seeing where they're recaptured, it gives an indication of whether they are moving between those areas. The traditional mark recapture tags are really good at understanding where fish move over a defined window, but they don't always tell us what they've done in that time. So, for example, if you were to tag a fish in the North Sea in the summer and recapture it in the North Sea in the summer, it doesn't mean it's been there all the time. It might have gone somewhere else. And so a lot of the electronic tags, they provide us much more detailed information.
So I just wanted to go into a bit more detail. Can we understand an individual's behaviour through these electronic tags, Serena?
Yes. So electronic tags can be very useful for understanding individual behaviours. So within a population, there might be very different decisions made on where they want to be and what temperature they experience, what depths they go to. All of that can be dependent on things like how old they are, maybe they're part of a different population. You know, their behaviour might need to change depending on if they're in the North Sea or the English Channel. And all of these things are quite important to get a better understanding of how adaptable they might be to things like climate change. That's something that we've looked into more recently. So some of the electronic tags will allow you to record depth at every 10 seconds if you wanted to, and temperature. So all of that can help to build a picture of what that individual experience is. When we get the electronic tag data back, we get very high resolution information on the experience in terms of depth, temperature, sometimes light, sometimes activity level. So that allows us to better understand vertically what they decide to do, whether that's by day and night, by season, and how that even changes by year. Sometimes we have deployments for 2 years, and that information can be really important to understand how consistent behaviour can be. We also can use the data that's recorded to understand where they're going on a day-to-day. So we can geolocate, which means work out the daily horizontal and vertical location between the point of release and the point of recovery. So to get that information, we have to do quite a lot of processing, which I won't go into detail about, but it uses information from the depth, the temperature, and relies on things like the satellite-derived products like sea surface temperature, temperature at depth, tidal signals. All of that can be used to better understand the location. Now, when we actually look at those day-to-day positions, that actually helps us build a picture of hotspots, where they like to aggregate. Are they feeding in a particular area? Are they spawning in a particular area? So that helps us to understand migration routes and how the fish behaves in their natural environment.
So it's not looking at the GPS signals, which can't go through water. It's much more processing, understanding those locations and those important pathways. Fascinating. So we've talked about quite a lot of information so far. So let's go through a couple of examples. What kind of animals can be tagged and have been tagged in the past?
But over recent decades, scientists at Lowestoft have tagged a whole range of marine creatures, including various types of shellfish, so crabs, lobsters, even whelks. A lot of fish tagging, so we've got a specialist team working with freshwater and what are called diadromous fish. These are microspecies that migrate between salt and fresh waters. So we've got a team actively working with species such as smelt, salmon, eel. In terms of fully marine fish, we've got, over the years, there's been a lot of tagging have either involved flatfish, so species such as plaice and sole, which are very, very important commercial species. Some of what are called gadoids, the kind of cod haddock that everybody will be very familiar with. And then various, what we call elasmobranchs, which are the shark skates and rays. So things like spur dog, thornback ray, species such as that.
We've also tagged a number of species of importance around the UK and overseas. There's been work on eels, so tagging both in the freshwater environments but also marine to try and understand migrations. There's a big question about eels, how they migrate to their key spawning ground in the Sargasso Sea, which is very far from UK waters. So there's been a lot of work on that in recent years and going over the last few decades. So we've had work on bass, which has included tagging from the North Sea all the way around to the Irish Sea to look at connectivity and behaviour. Some of these deployments have been out for 2 years and have had some really impressive data sets back. Finally, one of the biggest areas, I suppose, that has expanded in recent years has been tuna. So we've undertaken a lot of tagging work on bluefin tuna in the southwest, mark recapture tags, but also electronic tagging to understand the movements and migration of that very important species, which can reach in excess of 2 metres in length. We've also done a lot of tuna tagging work in the overseas territories, like St Helena, where we deploy conventional tags as well as electronic tags to feed into the wider picture of how tuna migrate and behave in the Atlantic. The information is not only useful for us in the UK and the overseas territories on their own to understand what their fish are doing, it also goes into wider understanding of these species.
Amazing. It's fascinating. I'm really enjoying this chat. So we've talked about first tagged fish that you ever have done, some of the examples that have been done over time. So what is next on your tagging wish list?
Well, over the years I've been fortunate enough to tag a whole range of different small dogfish, small sharks, various skates and rays. But the one that's really probably intriguing me at the moment is the stingray, a species for which the British Isles and Dutch waters are very much the northern limits. There's usually some little kind of hotspots for stingrays around the Channel Islands and in parts of the Thames. Wow. They tend to be found in very shallow water. And actually understanding the movement and connectivity of stingrays in Northern Europe, I think would be a really interesting study. It's on the wish list, but maybe a pipe dream.
And same question to you, Serena.
Okay, so maybe treading on Jim's toes, I'd very much like to tag a 6-gill shark. So one of the tuna that we tagged in St Helena, a yellowfin tuna, it It was predated and sank to the seafloor after about a month of deployment, and it looks like it was scooped up by a 6-gill. And the behaviour of that shark was incredible. So I would love to have a dedicated deployment on something that, migrates from 250 metres to 1,000 every day. This kind of very interesting and unusual behaviour, which I haven't ever explored.
Yes, I mean, the 6-gill shark is a fascinating species. It's been relatively well studied off the west coast of America, off Washington State, because they're actually found in much shallower water. So ironically, some of our better studied populations are not necessarily their natural habitat, but we do get 6-gill sharks in parts of the Celtic Sea and off the west coast of Ireland. So yes, so hopefully Serena will be lucky at some point.
Wonderful.
I just wanted to ask a question based on what you said there, Serena. How did you know it was eaten by a 6-gill shark? How could you tell that from the tag? Can you tell stuff about predators when the tag gets consumed, or can you explain that a bit more?
Yeah, sure. So when a fish has been tagged, you have a very characteristic profile that is specific to that animal. So a yellowfin tuna has this very consistent migration to deeper waters during the day, shallow at night. It's very linked to the diurnal cycle, so the day-night change. So you can very quickly build up a picture of what is a normal fish of that species. When it's predated, depending on what's eaten it, you might see a spike in the temperature. If it's eaten by a warm-blooded animal, like a mammal, you'll get a spike in the temperature. The profile will change because it's an air breather, so it will be more frequent dives to the surface. And so the actual profile themselves changes a lot. The reason why we thought it was a 6-gill shark, I spoke to a few specialists that have looked at 6-gill behaviour in the past, and knowing the habitat type around St. Helena and the species they've identified at that depth before, it was most probably a 6-gill. I mean, there's still a question mark, but it's something that, we'd expect it to be there. And the profile, the fact that it wasn't linked to the day-night cycle, it completely was just doing its thing. Didn't really care what time of day it was because it was not linked to the light levels. So that's why we thought it was a 6-gill.
Wow, fascinating. Thank you so much. It's just blown my mind. It's a 6-gill shark. That we've got stingrays in the UK, and that, 6-gill, such a deep water fish, is part of that kind of data analysis. Is incredible. Yeah, so let's think a little bit more about that technology and maybe the future of tax. We've talked a bit about the past. Let's talk a bit about the future, and we've talked a lot about electronics. So is electronics and the snazziness of that kind of innovation the future?
I think for electronic tagging, things are just going to keep on getting smaller, I imagine, and we're going to be able to collect more and more data points. That will allow us to be a lot more flexible with where we're tagging on the animal, making sure that we're minimising impacts to behaviour, ensuring that, the tags are as inconspicuous and not causing any behavioural changes. In addition to the tags being smaller, we imagine that there could be improvements in battery life. Rather than being limited to just maybe 2 years of deployment, we could push it and have, say, a 5-year deployment, 10-year deployment maybe, which would really improve our understanding of how these individuals are changing as they grow and as the environment is changing. I think as technology improves, maybe we just have more complicated or larger data sets that can be sent to the acoustic receivers, but I think the sky's the limit.
So is this all that needs to happen for doing more tagging work? Can we just rely on technology?
They shouldn't be viewed as being in competition. They're complementary. The traditional electronic tags provide invaluable information on the behaviour of fish, but currently the electronic tags, a few £100 each. A microcapture tag is a few pence. And so it very much depends exactly what you want to do. If you want to get fine-scale behaviour, you want to tag a few fish and and do everything you can to get those tags back. Any tagging program, if you really want to understand the ecology, the dynamics of a particular fish, it's using all tools available to us. There's so much of a greater interest now in the acoustic tags and acoustic arrays. So in parts of the English Channel now, there's several listening stations, and the scientific community is now working a lot more closely together to try to make sure that these listening stations are in broader areas. And then a tag that just emits a ping and can then be detected allows far more fish to be tagged, and then their positions can be basically triangulated. They can estimate where they are from the listening stations.
A lot of things can be complementary and help to build a stronger picture of what's happening. The conventional tag stuff, it's important to have that as a broad brushstroke, what's happening in the population as electronic tags, refine the detail of why and what they're doing in terms of their behaviour. The basic sample collection of, the sex and maturity of your fish, the sizes, the basic things that all need to be prioritised as well to help understand what happens to the population with a changing environment, with changing fishing pressure.
Thank you both. So we're getting to the end of our episode here, and so we usually finish with our feature. So for our fun fact today, can you reveal what kind of surprising secrets you've uncovered?
One of the beauties of working at a fisheries laboratory as opposed to a university is the continuity of knowledge and work. So we still reap the benefits of our predecessors' work. We've had mark identification tags for species such as spurdog and taupe that have been returned to us after the fish has been at liberty for over 30 years.
Serena, what would be your bit of information you want our listeners to take away from this session to get them excited about research?
I think some of the most exciting bit of work that I've been involved in is when you first download the tag data, you find out, sometimes that it's been predated. Some of the most interesting parts of the track can be the actual predation events, where in the water column it was predated from, trying to figure out what species it could have been predated from. So the most interesting bit isn't always the data that you've got back on the species that you've tagged. It's the data you get back on the species that predates that fish. There have been sperm whales, seals, ball beagles. There's been some amazing tracks that you get back. Allows you to see, an aspect that you didn't even imagine you were going to. So exciting.
Well, it's been a pleasure to speak to both of you today. Thank you so much for your time.
Oh,
thank you. Cheers, Kirsty.
Thank you for listening. This podcast was made by CFAS, the Centre for Environment, Fisheries and Aquaculture Science. To keep up to date with us, follow us on social media using @CEFASGovUK. We hope you can join us again next time.